Great. So hi, Austin.
Hello.
I'm here with Austin Argentieri, who is a brilliant researcher. We just wrapped up the Exposome Moonshot Forum, and I would really love to hear your thoughts about the meeting first of all, and then I want to ask you some questions.
Well, the meeting was really exciting. I mean, the biggest thing about all of this is just finally great to see some energy and some momentum around Exposomics, excitement about exposome science and coming to the roadmap of what we want to do together. But like the declaration of we will do it, we commit, I think is very, very exciting, very inspiring.
That was pretty energizing, wasn't it?
Yeah.
How did you get interested in exposomics?
My path to exposomics is pretty untraditional, I think. I started my career as an anthropologist and became interested in this idea of how can we start to map the social determinants of health.
Wow.
And then at a certain point we realized it's too difficult to choose just one or the other. And really, if we want to convince people in epidemiology and biology, in government, of the value of these types of exposures, we should think about what's the standard of evidence they want. So how can we make it anomic? How can we bring it to that sort of level of rigor and reproducibility and all these things? So we said, let's do an exposome-wide study instead. And my interest has always been in aging as well. So we said, well, let's do an exposome-wide study of aging and mortality. And then that was pretty much my entire PhD thesis, was working on that. So it all just started from there. Yeah.
So I was going to say the rest is history, but really because you're this very seminal paper that we're all very excited about because it draws the concept for all of us. Could you tell us a bit more about what you did in that paper?
Yes, so that paper was trying to map within just one cohort, the UK Biobank, the contribution of environment versus genetics towards explaining mortality and aging. People in the field of aging have been talking about for a long time. Aging is mostly not genetic, it's mostly not in your genes. Life expectancy has doubled in the last 200 years. That's far too fast for genetics to work. So it must be mostly environment. But no one had really quantified that. No one knew what in the environment. And we thought, this is probably something we can just test and quantify. So we looked in the UK Biobank. We carried out this exposome-wide association study to see how many different types of factors are associated with mortality and different aging traits, cognitive function, aging-related diseases, biological mechanisms, all this stuff. And then once you identify all of them, then you can do this cool thing if you can add them all together, like a kind of polygenic risk score, but a poly-exposure sort of model, and we can just quantify how much of mortality and aging-related diseases can we explain. And we end up sort of, I think, demonstrating what we had suspected, which is that, well, actually, if you put them all together and you compare them to polygenic risk scores and other things, you can explain nearly 80% in mortality by just age and sex in the exposome, and the influence is nearly 10 times greater than genetic predisposition. So I think it was received well, that paper, because it was validating to a lot of people in the sort of environmental health exposome field that there is a lot of merit to this idea that, like, if we have one place to invest, we'll get a lot more return for our investment trying to study these exposures than focusing all our attention just on the genomics.
I'm very biased, but I really do agree. Could you, if I'm not a genetics expert, right? I sort of know what polygenic risk scores are, but do you mind just explaining it in like simple terms for those who are not experts?
Polygenic risk score, just really simply, is you take all of the different genetic variants across the genome that are associated with a specific trait or a specific disease, and you combine them all, you add them all up, and then you get this single number that's an expression of what is your risk across all of your usually common genetic variants for that particular trait. So they're great tools, but they're usually not very predictive on sort of the tail ends of the distribution. People who are very high polygenic risk or very low polygenic risk, that tends to be meaningful, but on a whole, they don't explain a whole lot of the variation and a lot of outcomes that are complex and sort of polygenic.
Like multifactorial kind of, like we think. That is so cool.
So the way we did this in our study, because mortality is tough to get the genetics of, so we used this interesting approach where we took polygenic risk scores for all of the 25 major disease causes of death. So it was really, when we talk about comparing to genetics, we're talking about comparing to the genetic risk for most of the major disease-related causes of death in the UK.
That is so cool. And for, let's say, we know exposomics, and this field is very sort of multidisciplinary. There's so much expertise that comes in, right? Could you tell us a bit about your background in terms of like what did you study? How did you decide to go along that career path? You know, not all of it has to be like premeditated,
Yeah.
right? But like what was that trajectory and science that you learned to get you here?
Well, like I said, I started in anthropology.
Yeah.
I started doing qualitative research
That's so
on
cool.
sort of medical anthropology, different social influences on health and well-being. Then sometime around graduate school, I became really interested in biology and epidemiology, so I started working as a molecular epidemiologist first, and then I started my PhD, and I was there in a sort of big data institute in England, and so I was working a lot on computational biology and statistics and all these kinds of things. So I moved very slowly from qualitative to sort of quantitative and sort of big data aspirations
Big data,
and all of these kinds of things, but with the underarching interest that kind of unites all of it in understanding like the environmental influences or social influences on health. But at a certain point, I had a previous mentor who was very helpful for me in thinking through all these things, which is like, okay, we are convinced as social scientists that the social matters, that the environment matters.
Yeah.
But we need to convince the other scientists. And if we want to do that, we need to bring what we're doing to their standard of evidence, to their level of rigor, to their level of replication, all these things. And hence, like, the final step in translating to, like, let's do this as an omics, let's do it in, like, big data, let's move to the exposome.
That is so interesting. Was there like a clear point in time when you were doing all this incredible work as a molecular epidemiologist that you thought, okay, I really want to do a PhD now? You know, was there like a clear transition, or it was just like a smooth journey for you?
Oh, I always knew I wanted to go to
Yeah.
a PhD. It was just a matter of how quickly would I get accepted into a program. And so I only worked for a year or two in between master's and PhD. So it was kind of always just a bridge onto the next step.
So nice.
Yeah.
And my last question to you is, if you were to give, you know, young scientists these days, people training in the science or social or anthropology field, which is so cool, by the way, like advice on how do you even get started working your way towards something you're that excited about or passionate about, exposomics or otherwise, what would be like your biggest advice, you think?
Honestly, my biggest advice is find a mentor who believes in it and who will support you in doing it. I mean, I spoke with a lot of people all throughout doing my PhD who told me the work that we were doing was crazy or not useful or a waste of time or wouldn't produce anything or didn't just frankly understand the statistics of what we were trying to accomplish. But I had a mentor who was really supportive and like, if you think you have the idea and the goal, let's go for it. My mentor for my PhD wasn't an exposome scientist.
Yeah.
But, you know, she believed in this idea of let's not just cherry pick exposures, let's go big and try to do something big. And if we fail, we tried. And so we did. And so that was the most important thing for me. But, you know, equally, I came in with an idea that I was passionate about. And that's kind of the meeting of those two things is what gave us whatever success that we had.
I'm so glad. Thanks so much for sharing, and thanks for chatting with us.
My pleasure. Thanks for talking.
Yeah, thank you.